Hello everyone and welcome to today's live broadcast. The axolotl salamander is a model of nerve-dependent regeneration, presented by Johanna Farkas. She is a postdoctoral biologist at Northeastern University. I am Marjorie Torres of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located on the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button on the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Johanna Farkas. I will now turn the presentation over to her.
Hello everybody, and thank you so much for watching my presentation today. I'm Johanna, and I'm really excited to talk to you about the work that we do at Northeastern University on this amazing animal called the axolotl salamander. And I'll be talking to you today about the salamander's ability to regenerate and the ways in which we're studying this ability in order to hopefully unlock the ability to regenerate in other less regenerative animals. Now, when I say regeneration today, regeneration is a word that has a lot of different definitions depending on who you ask and in what context you ask. I'd like to define regeneration before we get started today so that we're all on the same page from the very beginning. And today, regeneration I will define as the return of a missing structure. So the example I'll be using today is the amputation of a salamander limb, which is then capable of regenerating back to a fully functional limb which looks identical to the amputated original. And this is an area of study that has become increasingly popular and increasingly well-funded because it has so many promising possibilities. So for example, this is an advertisement from the American Heart Association promoting the study of the zebrafish, which is capable of regenerating parts of its heart after they have been transected. And so we want to study this complete functional regeneration in animals that are capable of it in order to hopefully utilize their abilities in other animals such as ourselves. And the animal that I'll be talking to you about today is the Mexican axolotl, which us axolotl researchers like to refer to as the champion of regeneration. Might be a little self-indulgent, but these are really amazing animal models. I have here pictures of 3 different kinds of axolotls. So these are all the same species, but they have different colors, different pigments. On the right, you'll see that pigmented animal is a wild type, looks just like they're found in the wild. In the middle is an albino animal, and that animal does not have any pigment, just as we'd expect from an albino. And on the left is what we call a leucistic or white animal. And these animals have pigment in their embryos, they have pigmented eyes, but they are overall white, so we're able to actually see through them and do effective tissue grafts as well. And these are the sorts of animals that I'll be using throughout the presentation, these leucistic ones. Now, today I'll be discussing the regeneration of the axolotl limb. Again, if you amputate that limb, it grows back fully. You would never know it had even been amputated. But these animals have a vast array of other abilities that are really just incredible to witness and study. So for example, they can regenerate transected spinal cords. They can regenerate their gills if those gills are cut off. They can regenerate their retinas, their jaws. A number of internal organs are saying is sort of what doesn't kill them will just grow back. And while I'll be focusing on the limb today, I just want to talk a little bit about how these animals have such an amazing array of regenerative abilities, and they're just a really incredible model for this process. Particularly, as you'll see in the next slide, because they're a little bit unique in the animal kingdom. I don't want to make it sound like the axolotl salamander is the only animal that is capable of regeneration. There's actually a really surprising array of animals that can do so. However, they are somewhat more complex in terms of tissue and structure than many of the other regenerative animals that we study. So I have here an array of animals that are capable of tissue regeneration, ranging from the hydra at the top in the middle there, to frog tadpoles. So adult frogs can't regenerate very well, but tadpoles can, as well as zebrafish, starfish. In the bottom left is a flatworm called a planarian, and a crustacean known as an ostracod is in the bottom middle there. Now, what I hope you can appreciate in this slide is that the axolotl stands apart from these other animals because it's a little bit more similar to us. The axolotl is a tetrapod, meaning it has 4 limbs, much like we do, and it's also capable of regenerating more complex levels of tissue compared to a fish fin ray, which is not quite as complex as a fully amputated limb. Now, as you probably are aware, mammals are not particularly good regenerators, but there is one type of mammal, one genus of mouse called the African spiny mouse, or comis, which is capable of complex tissue regeneration. So these mice actually have this incredible adaptation where if a predator grabs them from the back skin will actually fall off, slough off, and then it will regenerate fully, up to and including the hair follicles, which is something we as humans are not capable of doing. So while regeneration is very rare in mammals, it is present in a small portion of mice, and study is ongoing to see how these mice resemble salamanders. And I will note that these mice have very recently been discovered and are still being studied for this ability. Now, limb regeneration in the Mexican axolotl is a very reliable process, and by that I mean it proceeds in a number of steps that we can really well characterize. The timing of this regeneration is going to vary depending on the size and age of the animal, but it always proceeds in the following fashion. First of all, the limb is amputated and the wound heals over, and this wound healing occurs incredibly rapidly compared to what we would expect from our wounds. So reepithelialization, just the covering of the wound site, occurs within 24 hours after the amputation is completed. After the wound has healed, what forms next is called a blastema, and that's the little bump that you can see at the end of the limb there. The blastema is a collection, basically a bag, of proliferating cells that forms from a number of different cell types. This can include fibroblasts, so connective tissue, and Schwann cells, which surround the nerves. It can include satellite cells found in muscles, and basically all of these cells go back in time a little bit and become like precursor cells, which then form this regenerating blastema and grow outward. And as that blastema grows, eventually the digits start to form, and over time, typically we could say about 30 days, but again this varies a lot depending on the size of the animal, we see differentiation of original tissue types, the formation of digits, and the conclusion of regeneration, which results in a limb that is functionally identical to the original one. Now, today I'm going to be focusing mostly on this blastema formation and growth portion of regeneration, because this is something that kind of sets axolotls apart from humans, right? We don't see blastemas, generally speaking, in human wounds, and it's also a big mystery. We want to know, how does this proliferating mass of cells form? How is it told to continue to proliferate? And how does it eventually form a new limb? So what I would like you to take away from this slide is simply the blastema definition as this idea of a proliferating mass of cells. And one really curious and fascinating, if you ask me, characteristic of axolotl regeneration is called nerve dependency. And this is a phenomenon that was first observed by Tweedy John Todd, who I swear was a real person, not a Victorian novel protagonist. It was first discovered by him in 1823, so this is a very old research question. And the axolotl itself is a very old animal model, having been studied since the 16 and 1700s. What Tweedy John Todd found is that if he intersected the sciatic nerve, and this is the nerve that leads into the hind limbs, he found that regeneration didn't take place. In many cases, the limb did not regenerate at all. In some, it actually deteriorated to the point that it essentially ended up back at the animal's body cavity. So this was a really big question, why, if he got rid of these nerves, and I'll show you how we do this in just a moment, why did this completely prevent regeneration? This is a topic that has fascinated us as biologists for nearly 2 centuries now. And this is how we denervate. So this is a bit more extreme of a surgery. This is simply opened up to show where those nerves are. But you'll see with the arrows, the 1, 2, and 3 are the 3 major nerves that head through the brachial plexus, which is found right about here in humans, and innervate that limb. So they start the spinal cord and they innervate all the way down that limb. what we can do is actually do a very small incision and find these nerves and just sever them. So we grab them with forceps and we just cut them. This means that we're not removing the nerve from the limb, we're simply cutting it, and this process completely prevents regeneration, so long as we cut at least 2 of those 3 brachial plexus peripheral nerves. Now, a normal regenerating limb looks like this. You can see the blastema is that little triangle at the end of the limb. That's around 11 days post-amputation. That's what DPA is going to stand for throughout this presentation. But if we denervate a limb at the day of amputation, if we cut those nerves at the brachial plexus, what we see is a limb that has completely failed to proliferate or form a blastema. The wound heals over just fine, but nothing happens after that. And in fact, you'll see some blood at the tip of that limb, indicating that there's something going on under the surface that maybe isn't usual. So what we can really say is that denervation, cutting the nerves leading into the limb, prevents limb regeneration at a very early step, right? So it prevents it essentially right after the wound has healed. After that, nothing will happen and the limb will not regenerate until those nerves come back in, which they do because the axolotl regenerates everything pretty much. Now here's an image of a blastema, an innervated blastema, and I hope you can see that really lovely bundle of proliferating cells at the end there. It's very dense. All those cells look pretty homogeneous, although they did come from different tissue types. But a denervated limb looks quite a bit different. So you may be able to see that this limb is essentially falling apart. There's a lot of osteoclasts in there, which are those big cells with multiple nuclei, and the limb itself is simply not proliferating at all. In fact, this limb is regressing and undergoing tissue histolysis instead. So this is a very drastic process that completely halts regeneration in its tracks for long lengths of time. And this is an immunohistochemical stain showing how intimately nerves contact that growing limb early on in the process. This is day 6 DPA, or days post-amputation, and the nerves are stained in red. And you can see that that nerve is innervating that growing blastema very, very closely. So it's growing straight into the wound site, it's heading into the epidermis, and a lot of proliferating cells, which are stained in green, that's a BRDU, a proliferative marker. There's a lot of proliferative cells sort of around that nerve, which indicates to us that the nerve might have some sort of role in supporting proliferation. Now, if this was a salamander-specific phenomenon, it would still be really interesting, and we'd still want to study it, but it might not be quite so relevant to other modes of health study. Happily, this is a phenomenon that's actually nearly ubiquitous in regenerating vertebrates, and in some invertebrates as well. So regeneration of the frog tadpole, which I mentioned earlier, is nerve-dependent, as is regeneration of, for example, the catfish barbel. Even starfish limbs do not regenerate if that oral nerve ring in the center is damaged. And we've seen this phenomenon in many other vertebrates that regenerate as well. Now, as I said earlier, and as you probably already know, humans aren't great regenerators, but that doesn't mean that nerve dependence isn't relevant towards our health. In fact, it's a very important question. And this is because nerve dependence is observed during human wound healing. As an example, paraplegic patients actually undergo impaired wound closure in wounds that occur in regions which are not well innervated or denervated. Also, peripheral neuropathy during diabetes can lead to what are known as diabetic ulcers. So what happens here are the peripheral nerves are damaged in the extremities, such as the hands or feet, and when that extremity is injured, there is no nerve present to support the healing of that wound. Thus, the wound, instead of healing over, ulcerates and becomes much more complicated from a health standpoint. And I have not included a picture of a diabetic ulcer because I don't know how many of you are eating right now. So what I'd like to take away from this introduction is that without an intact nerve supply, regeneration cannot take place. I'm going to say usually here because I'm about to show you some experimental exceptions. Now, like I said, this is a question that has persisted for almost 2 centuries, and it has been the source of study for quite a few scientists, especially in the past 100 years. One of these scientists was Marcus Singer, who conducted a lot of really beautiful experiments in the mid-20th century and eventually came up with what he calls the neurotrophic hypothesis. The neurotrophic hypothesis states that nerves supply the regenerating limb with a proliferative factor, by which I mean the nerves synthesize some sort of protein that is then released at the wound site and then promotes proliferation. So it's believed that this factor is synthesized up at the dorsal root ganglia, which are the cell bodies of the nerves that are innervating the limb. And from there, it travels down the limb and is released at the blastema to support regeneration. So this neurotrophic hypothesis has been very well supported, but the question of what exactly this factor or what these factors are has fascinated scientists for decades now, and it's a question that still doesn't have a really clear answer. Our goal for today, or our goal for the previous research as well, my goal for today is to show you the research that we've done on this topic, and we wanted to look at the possible identification of these factors. And we believe we should start in a really simple place. We should just find a factor that demonstrates three very, very basic characteristics. First of all, it should be found in the correct places, the peripheral nerves and the dorsal root ganglia, right? Because any factor that is supplied by the nerves should be found in those cell bodies and should also be found in the nerves and at the site of amputation. The factor should be capable of rescuing regeneration in denervated limbs, by which I mean we should be able to supply a denervated limb with this factor, and it should be able to grow at least a little bit, even in the absence of nerves. And inhibition of this factor should be capable of blocking regeneration, even in fully innervated, otherwise normal animals. And we decided to focus on a protein called nervigulin-1, which was a promising candidate because previous studies had been shown that it is upregulated after amputation, and that it may have to do with blastemal proliferation and growth. Now I have a very fancy animation here to show you how nervigulin works. Okay, maybe not that fancy. But the point is that nervigulin-1 is a ligand that's extracellular. It binds to a receptor called ErbB2, and then ErbB2 heterodimerizes with a receptor called ErbB2, which phosphorylates a number of downstream signals. And these signals have been found to be implicated in cell proliferation, cell survival, and cell migration. So you can see why it might be a really good candidate factor for looking at factors that promote blastemal proliferation. And there's a lot more I could talk about in terms of how this ligand or this protein works. But what I'd like you to take away from this is that nervigulin-1 is a very promising candidate, and its active receptor is called ErbB2. I'll be talking about those quite a bit in the upcoming slides. And our question, as I've said, is very simple. Does nervigulin-1 fulfill the three criteria that I described earlier? And this work has been very recently published. And first of all, we simply had to look for it. To this end, we collected tissues, in this case a dorsal root ganglion, again, those cell bodies at the entrance of the limb, and we sectioned and stained them for nervigulin-1 using immunohistochemical stains. So these are antibodies that tag the proteins of interest. So here in red is simply a tag that looks for nerve-specific cells, and then green is nervigulin-1, and you can see how beautifully expressed it is in these cell bodies. It is definitely there. It is also found in peripheral nerves. So you can see here we collected the nerves, we cross-sectioned them, and then we saw that nervigulin-1, again shown in green, is in fact present in the nerves. So these are two really good things to find. First off, it meant that the factor was located where it should be, if it is in fact a nerve-derived factor. And moreover, it is also found in the blastema. And here I'm mixing things up a bit. Nervigulin is now in red, whereas BRDU, the proliferative marker I mentioned earlier, is in green. And when we zoom in on this blastema, what we see is that, first of all, a lot of these cells are localized with nervigulin-1. You can see that they're stained in red. And also a lot of these proliferating cells are co-expressed with nervigulin-1. So many of these cells are both green and red, which indicates to us that there is at least some sort of correlation between proliferation and nervigulin-1. We can't say necessarily that nervigulin causes proliferation. We have to do many more experiments for that. But it is a sort of correlative relationship. And when we go in and actually quantify how many of these proliferating cells express nervigulin-1, it's somewhere, it is the majority of the cells, somewhere in the area of 60 to 70%. Now, of course, we found the ligand. We should also expect to find the receptor, ErbB2. We study ErbB2 because it is the active receptor of nervigulin. And in fact, here shown in red, you can see that this receptor is very strongly expressed in the blastema. So that's a relief. It's found where it's supposed to be. And moreover, if we actually denervate the limb, what we find is a lack of nervigulin-1 and ErbB2. So here on the left, I've shown you an ErbB2 stain in a denervated limb. And it's supposed to be in red. You can see there really isn't much ErbB2 to be found in this limb. But what's also interesting is all of these yellow cells, which you've probably noticed. Those cells are not, in fact, stained for anything. They're simply autofluorescing, so they show up as a kind of yellow or white color. And that is simply cellular debris, apoptose cells, red blood cells, just a lot of kind of chaos that occurs after denervation in these limbs. And this is something that we don't generally see in large quantities in innervated limbs. Now, when we go through and quantify how many cells express neuregulin-1 or ErbB2, we do find that, as predicted, given the images that you see here, that neuregulin-1 and ErbB2 are downregulated or lost upon denervation in a quantifiable manner. So that's number one. We found neuregulin-1 and we found its receptor. We can check that first characteristic off our list. Now, the second one was going to be a lot harder because to this end, we had to rescue regeneration in limbs that were denervated. So what we did here was we soaked these little beads overnight, and I've circled them there for you. These are tiny gel-like beads that can take up protein and then release it in a controlled manner later on. So this here is a control limb, and this limb was not denervated. It simply received controlled beads that had PBS in them, PBS being a saline that we use as a control in these sorts of experiments. Here is a limb that was denervated and then implanted with PBS-soaked beads. You can see that nothing happened as we'd expect in a denervated limb. There was no growth whatsoever. But when we soaked beads in neuregulin-1 overnight instead, we actually saw the growth of blastemas. So what is happening here is this limb has been denervated, but it's still developing a blastema because neuregulin-1 has been supplemented. We've added these little beads that are releasing it over time in a manner that we hope a nerve would in a normal, undenervated animal. And when we quantified these limbs, we were very excited and happy to find that, in fact, these neuregulin-1 treated animals grew quite comparably to the controls, whereas with the denervated animals that simply received PBS, those did not grow as all, as you would expect. So this is a fairly powerful method of getting these limbs to form blastemas and actually regenerate. Now, of course, there's always a question. Now, is it simply that neuregulin-1 is keeping the nerves alive and allowing those nerves to do whatever it is that they do in the limb? So we had to collect those limbs, section them, and stain them to nerve to make sure they were actually denervated. And I've shown here up at the top left your normal innervated blastema with those beautiful nerves stained in red. And here are what the denervated limbs looked like. You can see there's very little red, there are no nerves present, but there's still a blastema in that neuregulin-1 treated limb, implying to us that, yes, neuregulin-1 is capable of producing a blastema and proliferative growth even in limbs that have been denervated and do not have any nerve really whatsoever. And this was very exciting for us because this was one of the first instances we had where a protein proved capable of doing this in the Mexican axolotl. Now, it was really exciting. I mean, we had a blastema growing even when there weren't any nerves present. But our goal, in humans at least, is to grow back limbs, right? Not to grow back little tiny bumps. So we wanted to see if we could also get neuregulin-1 to produce digits later on in the process. So to this end, we added beads. We did the same exact protocol, but we did it later on after the blastema had formed. And if you denervate at this point, you won't see any growth. I'll show you a picture of that in a moment. But we saw that when we added neuregulin-1, we did in fact get the formation of digits. So they weren't well formed, they weren't exactly as beautiful as the control limbs were, but in comparison to the denervated limbs that only received PBS, they were really quite impressive and much larger. And we can see this when we quantify the growth as well. It plateaued a little bit over time. It seems neuregulin wasn't capable of really supporting that much growth late in the process, but those limbs did grow significantly more than the PBS-treated limbs did. So we can effectively cross off number 2 on our checklist and say that yes, neuregulin-1 does in fact rescue regeneration in denervated limbs. Now by comparison, this third thing on our checklist was in fact quite easy. All we had to do was inhibit neuregulin-1 to show that it blocks regeneration. So here again is an innervated control on day 13 with a lovely blastema growing at the end. And here is a limb that was treated with the very potent ErbB2 inhibitor. Remember, ErbB2 is the receptor. The inhibitor mubritinib, also called TAC-165. And treatment with this drug completely blocked regeneration. We saw wound healing, but we saw absolutely no formation of the blastema. We saw no proliferative growth whatsoever. In fact, if you took a look at this limb without knowing it was treated, you'd probably think it had been denervated. And this is borne out in the quantification of growth, which shows that there is very, very little growth in both denervated and drug-treated limbs. And this tells us that, yes, if we inhibit neuregulin-1 signaling, we are able to actually block regeneration, even in otherwise completely normal animals. And I'll also point out that our dosage in this case was 500 nanomolar, which is extremely, extremely dilute. So this is a very potent drug, and it is nevertheless very, very capable of blocking regeneration in these animals. Now, when we take a look inside these limbs, we don't see anything too surprising. Again, shown in green is the proliferative marker BRDU. The regenerating limb that has nerves is beautifully proliferating with lots and lots of BRDU expression. But that non-denervated limb, that drug-treated limb that has received ibrutinib, is not really proliferating at all. It still has nerves, which you can see there stained in red, but proliferation is almost completely absent from these limbs. They have been halted in their tracks, and they will be until the drug is removed. If you look down at the trichrome staining at the bottom, you could also see maybe just a little bit of blue at the tip of that drug-treated limb. And that is actually, this is a Mason's trichrome stain, so the blue is showing us connective tissue. And that is incredibly unusual in the axolotl because it indicates sort of the beginning of scarring. And these animals do not scar. They're not supposed to scar, even in epidermal injuries. It's nothing like humans where we form scars quite readily. And so this indicates to us that blocking neuregulin-1 signaling is doing quite a lot to these limbs. It's not just blocking regeneration, it's also causing some sort of aberrant fibrosis and perhaps having other unintended effects as well. Now, unsurprisingly, when we went through and quantified, counted the number of green cells that we saw, proliferation was very, very heavily reduced in these ibrutinib-treated limbs. Now, this was what happened if we added the drug at day 0, the same time that we amputated the limbs. If we add the drug later on, we got a bit of a different effect. So here's a control limb, 28 days post-amputation. We haven't done anything to it. And here is a limb that was denervated later on in the process, around day 16, after the blastema had actually formed. And what I hope you can see there is that this animal did form a limb. It's just a little tiny baby limb that has these little tiny digits at the end. And this has been known for a pretty long time among axolotl researchers, and it has suggested that the nerve is primarily involved in the proliferation of the regenerating limb, not in the patterning or digit formation of the limb. But we wanted to see, can we get this effect if we add the drug at that time instead of denervating? And in fact, we did. So again, this is a bit of an intermediate limb between the denervated and the innervated ones, but we did see the formation of these little baby digits, which indicate to us that the ibrutinib treatment, the ErbB2 inhibition, was blocking proliferation, but not the later stages of regeneration. And we can see this proliferative block if we take a look at the quantification of the growth, where over time, those drug-treated limbs simply plateaued. They got a little bit bigger than the denervated ones, but they did still just completely stop regenerating over time. And that fulfilled our third requirement. So simply to go over them again, we found that neuregulin-1 is in the peripheral nerves, in the dorsal root ganglia, in the blastema, exactly where you'd expect it to be, along with its receptor. We found that regeneration could be rescued in denervated animals if we added neuregulin. And we found that inhibition of neuregulin blocked regeneration in otherwise normal animals, which is all very exciting because it showed to us that neuregulin-1 has the characteristics that you would expect from an essential nerve-derived factor. Now we can ask ourselves, is neuregulin-1 the missing link between nerves and regeneration? Is that what the nerves are supplying that promotes growth of the regenerating limb? Now, I don't want to make it sound like neuregulin-1 is the only factor that is involved in regeneration. There's certainly a constellation of factors out there, and many have been studied over the years. These include FGFs, new anterior gradient, nerve growth factor, transferrin, insulin. It's a very, very long list. And it's believed that, of course, they're all interacting in some way. We don't yet know how neuregulin-1 interacts with these other factors, or which factors are upstream or downstream of which. But we are excited about neuregulin because we did find it in the nerves, and we believe it is a highly upstream signal coming from the nerves and signaling right into the blastema itself. And in future studies, we hope to explore this function of neuregulin, how it does what it does, and whether or not this function extends across other organs and systems in the axolotl. Now, I love this story. This was my doctoral work. I poured a lot into it, and I'm very fond of it. But as much as I do love it in neuregulin itself, it didn't quite feel like the whole story. And I want to show you why it is that I think that. So the denervated limb is a mess, to put it simply. There's just a lot going on, and a lot of it looks like cellular chaos. Right? So I showed you this photo earlier, which has a lot of osteoclasts, a lot of inflammatory cells. Here's another image of a denervated limb that's showing, again, everything in yellow is just cellular debris, and this limb looks like a mess as well. It doesn't look like a limb where you simply took away a positive proliferative factor. It looks like something else is going on here that's really messing up the regenerative process. And why might this be? Well, it could be because when you denervate a limb, again, you go in and simply cut the nerves at the brachial plexus. You're not removing the nerves from the limb itself. And a nerve that's been cut is a damaged nerve, and a damaged nerve is not very happy. In fact, nerves that have been exotomized and nerves which have been damaged release a lot of different signals, at least this has been found in mice and rats, release signals which bring in inflammatory cells and have all sorts of other effects. So a denervated nerve is not inert. It's exerting some sort of other possibly inflammatory effects on the limb. And that led us to ask, what happens when we denervate? Are we causing a sort of inflammatory environment that's going to block regeneration? Now, how much of this contributes to this phenomenon of nerve dependence? And so we had to look at this from a slightly different angle. Now, I want to show you a new model, or this is a model that's been around for a while, but this is new for the presentation. And it's a really fun and kind of interesting one. We couldn't look at amputation to start because amputation is a really complex process, right? You're cutting through nerve, you're cutting through muscle and bone and all sorts of different tissues. And we just want to look at what happens when you cut a nerve. So we use this model called the accessory limb or accessory blastema model, where you make a little tiny incision on the animal's skin. So you make a little wound, kind of just right here. And then you grab a peripheral nerve, so you cut it a little bit further down from that wound, and you redirect it so that it's sitting at the wound site. So what you end up having is this window in the epidermis, as well as this cut end of a nerve kind of just hanging out in that wound. And what happens is you actually get the formation of a bump. And that bump is pretty much identical to a blastema. So down on the bottom there, those are in situs that look for blastema markers. And these bumps produce pretty much all the same markers as a blastema does. So what this shows us is that all you need to form a blastema is a wound, again, a really simple one, and the cut end of a nerve. And we use this to our advantage to study whether or not nerves have inhibitory signals as well. So here are a couple of other diagrams of this model. You can see that we deviate that nerve, so we cut it, we grab it and move it, forms a blastema. Here's yet another example of how this works. And here's the kind of crazy thing that we did to test whether or not damaged nerves are capable of inhibiting regeneration, as opposed to simply removing a proliferative signal, which is what we've always thought of denervation as doing. So we did the accessory blastema study as we normally do. We made a wound, we deviated a nerve, and then we took a nerve bundle from another animal, in this case a GFP, or a green fluorescent protein expressing animal. And in the process of removing that nerve, we denervated it, right? We cut it off from its cell bodies. We then implanted this nerve bundle at the wound site of the animal which had undergone the surgery. So you can see they're fluorescing in green. That is a different bundle of nerves, one which has been denervated and effectively damaged. We placed it right next to that host nerve that should be producing lots of nice proliferative factors and causing this beautiful blastemal growth. And we saw that the GFP, the fluorescence, did not go down over time, which actually implies to us that those damaged nerves are still alive in some way. And when we sectioned these limbs later on, around day 15, we did see strong expression of GFP, indicating that yes, those cells are still alive, they're still doing something, maybe. Now, we wanted to see, were they in fact inhibiting the formation of this blastema? So here's a control experiment, very, very reliable production of these blastemal bumps. We had that in 16 of 21 of the surgeries that we did. But if we added this additional damaged nerve bundle, we only saw a bump forming in 2 of 12 cases. So in fact, when we added this sort of competitive damaged nerve to the normal wound site, we were able to completely block regeneration and bump formation. And if we take a look inside those limbs, we see a lot of cellular chaos. We see that the nerve has been deviated past where it should be. We see aberrant collagen deposition down there in blue, and we see a limb that is not very happy and not likely to start regenerating or forming a blastema anytime soon. Now, it could simply be that we're inducing this mechanical sort of inhibition because we're doing an experiment where we're shoving a lot of tissue into the limb. So to check for this, we decellularized those nerve implants by putting them in liquid nitrogen and also heating them up, so freeze thaw, freeze thaw. This got rid of the GFP, so it effectively killed off the cells, but the extracellular matrix and the structure of the tissue graft remained intact. And you can see that in the bottom photo here in the Mason's trichrome stain. When we freeze-thawed our tissues in this way, we did not see any inhibition of growth. In fact, we saw pretty normal blastemal growth altogether. And this indicates to us that it wasn't the surgery that was inhibiting the growth of these bumps, and it wasn't simply adding any sort of mechanical aspect of it. This was, in fact, the result of something active happening with these living cells. So perhaps these cells are secreting something that is competing with other positive signals at the wound site and preventing regeneration as we know it. Now, it could also be, you might be thinking to yourself, yeah, they're introducing foreign tissue into a limb. Of course, it's going to cause problems. And I should note that axolotls are incredibly receptive to grafts. They really don't reject them, not just from other animals, but in fact from other species of animals. So there have been experiments looking at grafts with tissue, frog and fish tissue, and the axolotls really don't reject those. But we did want to make sure that this was not necessarily something that happened with all tissues, right? So we implanted muscle into the limb site instead, and we saw that muscle implantation did not block blastema formation. In fact, we saw very lovely blastemas when we implanted this foreign muscle tissue. So it clearly isn't simply the result of other living tissue being present at the wound site. It's something that is at least moderately nerve-specific. Now, as I've said before, of course, our goal is not to grow bumps on human arms. Our goal is to grow back human arms. So we made sure to test whether or not this phenomenon was observed in regenerating limbs as well. And so to that end, we simply took peripheral nerve bundles, like we did for the previous experiment, and cut off the limb, and added those nerve bundles, either 1 or 2, right at the amputation site on day 0. And we did find that regeneration was slowed down, and in some cases quite messed up, which I'll show you in a moment, when we added these additional nerve bundles. So with 2 nerve implants, shown on the graph here, we saw a very significant decrease in growth. With 1 implant, we saw somewhat significant, but it wasn't nearly as striking. And of course, we did the control surgeries as well, where we freeze-thawed those implants. And so we still had the structure of the nerve present, but the nerves themselves were not alive. And these sham or freeze-thaw surgeries were almost completely identical to the controls, which did not receive implants as well, at all. So what we can say here is, once again, yes, there's some sort of activity coming from these damaged nerves that is in some way interfering with normal regeneration. And it really does interfere, which I'm going to show you here. So these are what the limbs that received 2 nerve implants look like. Remember, they're receiving damaged nerves that are giving off something, we're not sure what. And the sort of aberrations and abnormalities that we saw in these limbs, they were incredibly varied and really pretty bizarre and fun to look at. And these ranged all the way from blastema that was arrested very early on and simply never developed to digits. In some cases, we saw no digits or bifurcation of the arm. And even when digits formed, they were often either missing 1 or 2, or the digits themselves were quite abnormally shortened, which we can see in this last image here. Now, when we took a look inside these limbs with Alcine blue staining, you can see on the right there that there's a lot that's wrong in this limb. It only has 2 digits. It has what looks like maybe a carpal floating off into space, as opposed to our control and our sham surgery limbs, which are beautiful, fully formed, and look almost identical. So again, there's something happening here. When we introduce a damaged nerve into the environment, we are in some way competing with the nerves that should be producing something proliferative. And I'll show you how this all wraps up together in just a moment. Now, we are able to score these limbs, so what we can do is go in and actually say whether or not these limbs fully regenerated and kind of classify them based on what we see. So here in green, we have limbs that were ranked or scored as a 3. That simply means that they regenerated fully and perfectly. That's what you want to see. They look just like the original limb. The yellow, or score 2, is a limb that was highly abnormal in its morphology but did produce some sort of digit. And the score number 1, that's shown in red, is a limb that simply didn't grow at all. It maybe formed a little tiny blastema, but it never progressed past that. And what I hope you can see in this figure here is that the denervated limbs mostly just didn't grow. But the limbs that received damaged nerve implants, the ones that we did that sort of weird grafting surgery into, those limbs ended up looking really, really bizarre. They had a lot of abnormalities. Some of them didn't grow, some of them grew perfectly, but as a whole, the majority of them showed some sort of strange in-between aberrations. And we're working now to determine what are these factors which are produced by these nerves and eventually result in these aberrations, and how does it interact with Nerve-1? Is it in any way involved in this process? So the conclusions that we've come to so far are that limb denervation is a little bit more complex than it might appear at first. So we believe that there are sort of two major aspects to limb denervation that combine in order to prevent regeneration of the salamander limb. So first of all, when you denervate, when you go in and cut those nerves at the brachial plexus, you are losing proliferative factors that are released at the wound site. So for example, nerve-1. We know that nerve-1 goes down after denervation. We know it isn't able to truly act as it should. And so we can say that half of denervation involves the removal of a positive proliferative signal. We're preventing that signal from traveling from the cell bodies down the nerves and from being released at the wound site. But we can also now say that there's a second aspect, a little bit less appreciated, perhaps less studied, to limb denervation that also prevents regeneration. And that is this sort of introduction of a negative or inflammatory signal from those nerves themselves which have been denervated. So what we can say here is that denervation produces a lot of very damaged nerves that are giving off some sort of factor that is anti-proliferative, potentially. Or they could potentially be bringing in leukocytes and white blood cells to prevent regeneration in other ways. And this is something that we're still working to determine. Is it a direct inhibition from some factor released by damaged nerves, or is it that they're causing inflammation that then in its own way prevents regeneration? But we can thus kind of summarize all of this by saying that denervation of the axolotl limb prevents regeneration in two ways. It removes a proliferative signal, a positive signal, and it adds an inhibitory, perhaps inflammatory signal. And our dream is to one day find the ways in which these two halves of denervation interact with each other and perhaps unite them to get a whole, complete picture of what happens after denervation and how regeneration is affected by it. Now, other areas that we're excited to explore, first of all, we'd really love to see how Nerve-1 affects other organs and tissues in the axolotl. So these are just a couple of photos. They're very, very kind of preliminary and rough. But here you have a heart on the left that's simply a developing axolotl heart. And on the right is a heart that's been injected with Nerve-1. And you can see that proliferation has gone up quite considerably in this heart. So we'd really like to explore whether or not Nerve-1 is involved in the regeneration of the axolotl heart, as axolotls do have the ability to recover from cryo-injury and transection of the ventricles. Nerve-1 is also being studied quite extensively in mammals for its heart protective and potentially regenerative ability. There's some very exciting recent studies showing that it may, in fact, be capable of at least inducing some sort of cardioprotective effect in mammals. And we're excited to explore this in the salamander as well. And also, because this is such a flexible and fascinating and highly, really quite a variable model that we can do a lot of fun experiments on, we'd also like to use it to look at other aspects that maybe haven't really been thought of previously in terms of regeneration. So for example, when you cut off a limb pretty proximal to the animal, that animal has to regenerate a joint. And this provides us with a really great model for looking at joint morphogenesis and formation, because studying joint morphogenesis in the developing animal is often quite difficult. They're usually very small and often protected by a fetus or by a uterus. So what we can actually do here is amputate an axolotl limb and then study how this joint forms as it is regenerating. We can do all sorts of really fun manipulations to that regenerating limb to see how does immobilizing the limb affect joint formation? How does reversing flexion of the limb change joint formation? And this is something that we'll be working on going forward in my postdoctoral work here at Northeastern University. Very excited to see where it takes us. And with that, I'd like to thank all of these people listed on this slide, members of the Monaghan lab, the Whited lab, our collaborators, the Shefelbine lab, which I'm now doing postdoctoral work in, our funding sources, and of course all of you for watching today. I really appreciate that you did. Thank you so much, and I'm happy to take any questions.
Thank you, Dr. Johanna Farkas, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, can axolotls die of old age, and do they get cancer?
Okay, so axolotls, you might think that because they're so amazingly regenerative, that they might be capable of living forever, perhaps. But in fact, they do die of old age. Now, they live a little bit longer than you might expect from a salamander, about 20 years or so, but they do, in fact, senesce over time, so they grow older, and they do eventually die from old age. Now, one thing that's really amazing about salamanders is they retain their regenerative and their reproductive abilities throughout adulthood, which is something that sets them apart from many other vertebrates. In terms of cancer, this is another area of axolotl research that's just really fascinating and there's a lot of potential in. Despite the fact that many of the pathways, the genes and proteins they utilize are oncogenic, so that, for example, ErbB2, which I talked about, is highly implicated in some cancers, they are highly cancer-resistant animals. So axolotls do sometimes form neoplasms in a very small percentage of animals, but as far as metastatic cancer goes, it's very, very, I can't say for sure that they are immune to it, but it is extremely rare to the point that it hasn't really been characterized in these animals. And moreover, attempts to actually induce metastatic cancer in axolotls have not to this point succeeded. So going forward, one major question is, how is it that they don't seem to get cancer at the rates that we do, and how are they able to control these proliferative oncogenic pathways so tightly such that they don't accidentally get a tumor when they are regenerating a limb?
Thank you for that answer. Our next question is, will an axolotl limb grow back if you amputate it more than once?
So you might wonder, now, is the axolotl limb capable of actually just continually regenerating? And in fact, it is. It's a really remarkable feature of the axolotl salamander that you can amputate a limb, and you can amputate it once it's grown back, and the limb that you'll see is pretty functionally identical to the very first limb even. And this can be repeated over and over and over again without any loss of regenerative fidelity. In fact, some recent research has shown that these repeated amputation limbs are in fact really, really close to the original. Now, there is one exception to this, which is if you amputate a limb bud very early on, so for example, a blastema that forms, if you amputate that repeatedly, it seems that somehow the pool of regenerating cells is depleted or exhausted in some way, and you get just a little tiny baby limb instead of a fully functional regenerated limb. So we can say that it is capable of regenerating over and over if you cut the fully regenerated limb, but we still don't exactly know how similar the first limb is to the, or how similar the original limb is to the, say, 50th regenerated one. We do know that it looks and functions the same, but in terms of what's actually found inside of it, that's still an area of study that is being looked at.
Okay, it looks like we have time for one more question. Do humans share many genes with axolotls? Is NRG1 found in humans?
So yeah, that's a great question because I've talked a lot about NRG1 and its pathways and its abilities in the salamander, but would it really matter if humans do not, in fact, express this gene? But the good news is that NRG1 is not just found in humans, it's found in a huge variety of animals, down to very, very early primitive invertebrates. So NRG1 is very well conserved, we'll say, across phylogeny, and really it's the abilities of NRG1 that are simply different from animal to animal. So it is possible that someday we'll be able to utilize NRG1 in some way in humans to have cardioprotective effects potentially, to help grow limbs back. But again, we always have to worry about the fact that its receptor can be oncogenic. Now, humans and axolotls do have a surprising number of conservation between their genomes. Axolotls have a much larger genome, same number of genes, but about 20 times the size is their genome because they have so much what looks to be junk DNA. And while there are many salamander-specific genes, and of course we aren't closely related, they are still, as I said, vertebrates that are sort of the closest to us that are capable of this complex tissue regeneration. And in looking at how they're able to regenerate and which pathways and genes they utilize, we can hopefully see how humans might be able to do the same as well. And we're comforted by the fact that many of these major genes, such as NRG1, are in fact shared between humans and salamanders.
I would like to once again thank Dr. Johanna Farkas for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us, and we hope to see you again soon. Goodbye.